Okay, before we begin, are there any questions? Guess I should shut those down. All right, not hearing anything. So can somebody confirm you can hear me?
Confirming.
Thank you. Let me see. Share my screen here. Should be able to see my screen now. All right, today is February 3rd. And I think I have an appointment tomorrow, but I'll have to check. And we're supposed to be on chapter 4. We should try and finish chapter 4 today. There is a lab today. Any questions about what we're going to be doing? You might note there is no quiz today. Not this week, I mean. I haven't talked today. My voice seems a bit hoarse. Let me get a little something to drink here. All right, let's go to chapter 4. Did we talk about endospores last time? Yeah.
Um, I think we did a little bit. Yeah.
Okay, thank you. Yeah, that's what I have, that we had talked about endospores. And did I show you the movie on endospore formation?
Yeah, the short YouTube clip.
Yeah, it's not a real good movie, but it's a little
Yeah.
video on endospores farming. All right. So remember, endospores are a survival structure. One cell produces one endospore, and then the parent cell that makes the endospore dies. So that means you only have one cell. It is not reproduction. It is a survival strategy. It's not a reproductive strategy. Any questions about any of that? All right. So we're going to briefly talk about eukaryote cell structures. Here on the right side, you're seeing an animal cell. Let me see if I can blow this up. Now we have the plant cell on the left side sort of cut off. Let me see if I can move it over. I can't. Maybe shrink it down a little. Whoop, that's too far. That's odd. This computer works a little differently than most others. No, that didn't work either. Oh well, I don't seem to be able to shrink that down any. Maybe I can go here and just blow it up. Sort of missing it there, but
It looked like that slide before you had adjusted it. It looked like it had been cut off the screen. That might be why it was off.
no, there's something on the right
Oh,
here. I can show you if I scroll over. There you go. It's just not totally showing. So we're seeing a plant cell on the left and an animal cell on the right. The difference is a plant cell has a cell wall around it. The cell wall gives it a shape. So this one would be like a hexagon. The animal cell only has a plasma membrane, and so it will be whatever the plasma membrane tends to be, which tends to be roughly ovoid or circular. You do see some organelles, and actually most of them, most of the organelles you do see in a plant cell and an animal cell, such as the nucleus, the mitochondria, right there and right there, the Golgi apparatus, also called the Golgi body. You can find them in both plant and animal cells. The flagella, or flagellum here, it does tend to be found in more animal cells than in plant cells, but you can find a flagella or flagellum in a plant cell, like the gosh, is it the sperm cell? Or no, it's the pollen cell of a oh, what is that called? A fern cell. The pollen cell of a fern has a flagellum. There's a few other plant cells that have flagellum. It's not as common. In animal cells, there's only a few cells, at least in humans, that have um, flagellum, and the number one would be a sperm cell.
I think the book said the only cell in a human that has a flagellum is the sperm cell.
You could be correct. I was thinking the throat cell, but now that I think about that, that's cilia.
Cilia. Yeah,
Yeah,
that's cilia.
yeah. Like I said, there's only a few animal cells. Well, in humans, only a few animal cells that have the flagellum, and like I said, the sperm cell is one. Um, and there might not be others. I don't know. There are a few organelles we only find in plant cells, like a chloroplast. The central vacuole is found only in plant cells, although you can find it in like a protocell, but you do not find it in an animal cell. You can, however, find a vacuole in an animal cell. It's not shown here, and that's the central vacuole there. The central vacuole is the largest organelle in a plant cell, larger than even the nucleus. What's an organelle you can find in an animal cell, but you cannot find in a plant cell? The microtubule, no, not the microtubule, what this? The centrosome is an organelle. It's a non-membrane bound organelle that you can find in an animal cell. It helps make the mitotic spindle. Plant cells have a mitotic spindle, but they do not have a centrosome, including the centriole and the paracentral material. When I'm talking about organelles, you should know that usually I'm referring to a membrane-bound organelle where there will be a membrane around the organelle separating the organelle from the rest of the cytoplasm. Or in the case of the nucleus, that is an organelle, that will separate the cytoplasm from the nucleoplasm. But I could mean a non-membrane bound organelle, such as the flagellum shown here, or the centrosome shown here, or the microtubules. These are non-membrane bound organelles. The chloroplast is a membrane bound organelle only found in plant cells, not found in animal cells. The mitochondria, a membrane bound organelle. The Golgi apparatus, a membrane bound organelle. Any question about the difference between a membrane bound organelle and a non membrane bound organelle?
So membrane bound just means like they have their own membrane
Yeah, it
surrounding
has
them.
its own membrane that separates the boundary of the organelle from the cytoplasm. In um, in bacteria and archaebacteria, we do not have any membrane-bound organelles. In eukaryote cells, you do have membrane-bound organelles. The membrane-bound organelles divide up the cell into different compartments, which literally is a different sac. And usually the membrane-bound organelle, if not entirely, they perform a different function for the cell. Like the mitochondria makes ATP, the what was that membrane-bound organelle there? The peroxisome, I don't remember what it exactly does. I think it engages in oxidation, but the where is that here? Looks very the lysosome looks very similar to the peroxisome. Uh, engages in lysis of some material that is usually brought into the cell, and then the lysosome fuses with the phagosome to dissolve the contents of the phagosome. So the lysosome helps dissolve or lyse the organelle or something brought into the cell. The chloroplast engages in photosynthesis. So usually the organelles perform a specific and a different function than another organelle. Any question about any of that? All right, so the eukaryote cell is divided into different compartments that we call membrane-bound organelles. The membrane-bound organelle performs a different function than another membrane-bound organelle. One possible exception is a central vacuole does perform many functions. It can store components the cell wants to store, and then it can also store waste products. So imagine this being your refrigerator where you're storing your food, but then you're storing your waste products. Instead of going to the toilet, you store it in the central vacuole. And that's just the way plant cells are. The smaller vacuole, which is not real large, like the central vacuole, stores things that the cell is storing. And the difference between a vesicle and a vacuole is a vesicle is a membrane-bound organelle that is moving components from one region of the cell to another region. Once the vesicle stops moving and then starts storing the contents of the vesicle, we don't call it a vesicle anymore. We call it a vacuole. And we'll talk briefly about that in a little bit. Any question about any of that? All right, this is actually an electron microscopic image of an animal cell on the left and a plant cell on the right. Let me shrink that down just a little so we can see all of it. Shrink that down just a little bit. Maybe I can blow this one up. Yep, I can. All right, so we're seeing the difference between a prokaryote and a eukaryote cell. Difference in the size. A eukaryote cell is about 10 times larger than a prokaryote cell. And this is actually showing you close to the average size of a bacteria and down to the smallest size. The smallest prokaryote cell we can find is about 0.2 micrometers in diameter. However, it's thought that those cells are non-viable, that the largest or the smallest viable cell is about 0.4 micrometers, and then the up to the average diameter of about 2 micrometers in diameter. We can get bigger than that, and that's really about the smallest we can get. And then the largest cell is the chicken yolk cell, and it's much larger than 100 micrometers. There is no nucleus in a prokaryote cell. There is a nucleus in a eukaryote cell. Membrane-bound organelles are absent in prokaryote cells. They're present in eukaryote cells. Both of them may contain a flagellum or a flagella. There are some differences. We're not going to really talk about the differences in the flagella. The glycocalyx can be present in both eukaryote and prokaryote cells. It's a substance secreted by the cell external to the cell. The cell wall, if it's present in a prokaryote cell, is complex, typically made in bacteria of peptidoglycan. In archaea, you don't need to know about it. It's a pseudomurine, similar to peptidoglycan, but it's a different molecule. And if it's present in a eukaryote cell, the cell wall is chemically simple, like cellulose is the main component of the cell wall of a plant cell and certain protists. Animal cells, of course, don't have a cell wall. Plasma membrane, generally they lack sterols in bacteria and archaebacteria. That isn't true if the prokaryote cell wall lacks, the prokaryote, cell lacks a cell wall. It does have sterols in it. And animal cells, the plasma membrane has a sterol in it. You all know the sterol is cholesterol. And the cell plasma membrane does have carbohydrates in it. Generally, the plasma membrane of a prokaryote lacks carbohydrates. The cytoplasm, this is actually wrong, saying that there's no cytoskeleton. The cytoskeleton now has been known for about 15, maybe even 20 years in prokaryote cells. However, the cytoskeleton is simpler in a prokaryote cell than in a eukaryote cell. So the eukaryote cytoskeleton is complex. The cytoskeleton is a network of proteins running in the cytoplasm of the cell. And the cytoskeleton is much more complex in a eukaryote cell than a prokaryote cell. There is cytoplasmic streaming. I'll show you that in just a little bit in a eukaryote cell where the cytoplasm actually moves around. There is no cytoplasmic streaming in a prokaryote cell. The ribosomes differ in a prokaryote. They're smaller, 70s in size. They're larger in the eukaryote cell. They are 80s in size. That differs for the ribosomes and the mitochondria and the chloroplast, and we'll talk about that later. The chromosome in the DNA of a prokaryote cell, at least a bacteria, it's usually a single chromosome. That chromosome is circular. The chromosome lacks histone proteins. In a eukaryote cell, the chromosome is multiple chromosomes. They're linear chromosomes, meaning there is a separate beginning place and an ending place. And the chromosome, the DNA is wrapped around the histone proteins in the chromosome. Cell division also differs. Bacteria divide primarily by binary fission, and a eukaryote cell divides by mitosis. Any question about any of this? All right. If a bacteria cell can move or swim, it has a flagella because that's the only structure a bacteria cell has to move. However, eukaryotes have three structures to move. They have flagella, so that's a possibility. The sperm cell has one. There's cilia, which is shown on this paramecium. There's also cilia in our human throat cells, which can, well, it makes the paramecium move. In the throat, it actually makes the mucosal lining move. And then the third structure a eukaryote cell has for moving is pseudopods. A white blood cell can move by moving out the cytoplasm in one direction of the cell, forming a pseudopod, and then moving in the cytoplasm on the opposite side so the cell moves forward. Any question about that? A difference between the flagella is there's few of them on a cell. Even if the cell has multiple flagella, there's few compared to the cilia, and the flagella are long, whip-like structures. The cilia, if the cell has it, it's many. They tend to beat in unison, and they're short, much shorter than the flagella. All right, I think that's all I want to talk about eukaryotes, with a few exceptions. Let's take a look at cytoplasmic streaming found in eukaryotes where the cytoplasm actually moves. This might take a little while to load. Well, thought I had it. There it goes. After
Switch
a
to
commercial,
TurboTax and file your own taxes free in the app if you didn't file with us last year. Just
shot in real time. There, you can see the chloroplast moving in the cell from the cytoplastic streaming. Can't see the central vacuole, but in this part of the cell, the chloroplasts are moving around. All right, any questions about cytoplasmic streaming? The cytoskeleton, it's the structure of proteins in the cell. Let me see if I can blow this slide up. So here we can't see the cell membrane, but it's around right here. And that is actin filaments, which are part of the cytoskeleton, one of the proteins making up the cytoskeleton. There's three parts to the cytoskeleton: microfilaments, intermediate filaments, and microtubules. Here we can actually see this cell right here, probably right there, and that's probably another cell right there. And there's the nucleus standing blue, which is actually lacking the cytoskeleton. It's just standing blue for some reason. But this one is showing actin filaments in red, microtubules in green. The nucleus is in blue, which is not part of the cytoskeleton. Remember, the cytoskeleton is just a framework making in an animal cell, kind of the cell structure, meaning the cell shape and structure, but in plant cells, the shape is more dependent on the cell wall. And in bacteria, the shape is not dependent on the cytoskeleton. The shape is dependent on the cell wall. I'm skipping this because you've had this already in a preliminary course, and I mentioned earlier that I would largely skip the eukaryote functional anatomy. Let's briefly talk about the endomembrane system. Eukaryotes have an endomembrane system where various organelles work together to make and move molecules around in the cell. Let's see if I got that there. Yep, here we go. So the proteins initially start from the largely the rough ER, although they could start on any ribosome. Remember, a rough ER is simply just the ER with ribosomes bound to it, making the ER look rough. The smooth ER makes lipids, but the rough ER makes proteins. Proteins tend to be made in the rough ER, and the proteins bud out of the rough ER, where a vesicle actually is made and buds out of the ER, and then transport that protein inside the vesicle to the Golgi apparatus. The Golgi apparatus, it binds to the Golgi apparatus on this side of the Golgi apparatus. The Golgi apparatus then modifies and may chemically change the protein. For example, I did mention that if all proteins start, at least in eukaryotes, start with methionine, if you don't want that protein to start with methionine, it needs to be removed, and that methionine will be removed in the Golgi apparatus. The Golgi apparatus tends to methylate proteins and do some other things to proteins, but the Golgi apparatus modifies the protein. And then on the other end of the Golgi apparatus, it buds out a vesicle, which then transports the protein to where the protein needs to be. It could be transporting the protein to the cell membrane, where the vesicle will actually fuse to the cell membrane, and then the proteins will be dumped out of the cell. And this happens in a teenager with raging hormones, where the Golgi apparatus makes the hormone modified, not the Golgi apparatus, sorry, the endoplasmic reticulum makes the hormone, the protein, and is taken to the Golgi apparatus by a vesicle. The hormones can be modified, and then they are made into another vesicle, which then has it fuse with the cell membrane, and the raging hormones of the teenager get expressed. Any question about any of that? If, on the other hand, the vesicle just stops moving and starts storing the proteins for later use in the cell, we no longer call that a vesicle, we call it a vacuole. And then the proteins will be stored until they're used by the cell. And the vesicle will just store it someplace in the cytoplasm. Where the vesicle is moving, it's called the vesicle. Where it stops moving and starts storing, we call it a vacuole. Any question about any of that? From the smooth ER, you can have a vesicle bud out. So the smooth ER is making lipids, and that vesicle can bud off of the smooth ER and then go fuse with the Golgi apparatus, and the lipids in the vesicle will undergo the same process as the proteins, meaning they'll be modified by the Golgi apparatus, which will then form a vesicle budding out of the other side of the Golgi apparatus. And the vesicle will then move to the correct location in the cell, and it could just end up being stored, in which case the vesicle will be called the vacuole until the contents are used. But the vesicle could fuse, say, like with the nucleus, and then the lipids will be delivered to the nucleus. Any question about any of that? Anyways, that's the endomembrane system involving the making of a protein, generally at the ER, or the making of a lipid, also the smooth ER, and then the vesicle transporting it to the Golgi apparatus, the Golgi apparatus modifying the contents, either the protein or the lipid, and then the Golgi apparatus budding off in another vesicle, bringing the content to the correct location, or just storing it, in which case the vesicle will be called a vacuole. Any question about the endomembrane system? All right, let's talk briefly about the mitochondria and the chloroplast. These organelles differ from other membrane-bound organelles because they have two membranes, an inner membrane shown here and an outer membrane shown here, at least of the mitochondria. The chloroplast has the same, although its inner membrane is much more complex and much more folded. The mitochondria and the chloroplast also differ because it has ribosomes inside the matrix of the organelle, either the chloroplast or the mitochondria. And these organelles also have a DNA molecule inside them. Not shown here, but someplace in the matrix is a DNA molecule. Any question about any of that? I should mention the ribosomes of the chloroplast and the mitochondria are smaller than the ribosomes of the rest of the eukaryote cell. The ribosomes of the mitochondria and the chloroplast are 70S in size. The ribosomes of the rest of the eukaryote cell are 80S in size. Any question about any of that? They're showing you the outer matrix of the chloroplast, and there's a portion of the inner matrix of the chloroplast. All right, let's talk about the endosymbiotic theory. This is a theory to explain the evolution of how eukaryote cells obtained a mitochondria and a chloroplast. I think I will stop here and open up Paint. Well, I'm not seeing Paint here. Just a minute. Oh, there it is. A long time ago, we had a proto-eukaryote cell, which was a big cell, and it could only make ATP from fermentation. Taking one glucose molecule to make 2 ATP. Oop, do that right. And this was a proto-eukaryote cell. It wasn't quite a eukaryote, but it was a larger cell. And this larger cell engulfed bacteria. small bacteria. We'll make basically a rod. How the cell engulfed it was by phagosomes, which came out of the cell. And grew larger. Until they finally touched, and then the phagosome membranes fused. And then this bacteria cell was now inside the proto-eukaryote. For some reason, whatever it was, the proto-eukaryote did not dissolve and eat the smaller bacteria. It just kept it around and kept it for use for probably digesting later, and it never digested it. And what happened was that glucose moved from the proto-eukaryote into the bacteria. Now, the bacteria had aerobic respiration. To make ATP. So one glucose molecule could make 38 ATP. Oops, need a space in that. And for whatever reason, probably because life was young, the glucose just moved into the bacteria, and then the ATP that the bacteria made, some of it moved into the proto-eukaryote. The proto-eukaryote was trying to figure out what happened because it lost one glucose molecule and it gained many ATP, many more than two, probably not 38 because the bacteria probably saved some of the ATP for itself. But even if it was only, I don't know, what's half of 38, 19? If it was only 19, that's quite a bit more than 2 ATP that the proto-eukaryote could make. And the proto-eukaryote sort of figured out that it was getting all this ATP from one glucose molecule from the bacteria, which was sharing its ATP with the proto-eukaryote. And so the proto-eukaryote said, this is a good deal. I can only get 2 ATP per molecule of glucose. If I have this bacteria inside of me, I'm getting many more than 2. Let's say at least 19. It might have been higher. We don't know the exact number. It could have been approaching 30 or even maybe even approaching 36. But whatever it was higher than 2. And so the proto-eukaryote started keeping the bacteria inside of it and just fed it glucose. And then the bacteria gave the ATP, or at least some of it, to the proto-eukaryote cell. And so the two started living in symbiosis. After time, we now only find the bacteria inside the proto-eukaryote. And the proto-eukaryote has had this bacteria inside of it for so long that we no longer call it a proto-eukaryote. We call it a cell, eukaryote cell, with an organelle in it that was once derived from a bacteria. And this organelle we call a mitochondria. That is the theory of how eukaryote cells develop their mitochondria. what proof do we have for this theory? First, you'll note that this organelle, the bacteria, has two membranes, its own cell membrane right here, and then the organelle membrane of the protoeukaryote when it first took in the bacteria, meaning it was brought in by an organelle. And the mitochondria do have two membranes. If we look at the ribosome, well, first of all, there's DNA in this cell, and we would expect DNA in that cell if it was a bacteria. And if we look at the ribosomes of a eukaryote are is it 38? Sorry, I don't remember the so the ribosomes of a eukaryote are 80S in size, and the ribosomes of a bacteria are 70S in size. If we look at the ribosomes of the mitochondria and the chloroplast, they are 70S in size. Oops, don't need that anymore. There we go. If we look at the DNA cells, DNA, we find the DNA of the mitochondria and the chloroplast. It's a single circular chromosome, and the DNA of a eukaryote cell is multiple linear chromosomes. Go back to my slide here. If we look at some protists, there are a few amoeba which lack a chloroplast. What these amoeba do is they engulf a bacteria and then use the bacteria to perform aerobic respiration. Let me see, it's amoeba feeds the bacteria glucose, and then the bacteria makes ATP with aerobic respiration and shares the ATP with the amoeba. So there actually are some simple protists that lack a mitochondria, and to survive, or at least to help them survive, they engulf a bacteria and then use that bacteria similar to a mitochondria. Here we're looking at a paramecium, which is actually engulfed in algae, and the paramecium is giving the algae a home, giving it light and giving it CO2, and then the algae is taking the light and the CO2 in the water to make carbon dioxide, not make carbon dioxide, to make glucose from carbon dioxide, and then it shares the glucose with the paramecium. The chromosome of the mitochondria and the chloroplast, I already mentioned, it's circular and there's usually one. And that's similar to the bacteria, differ from the chromosome of the eukaryote cell. We find no histones in bacteria cells. We do find them in the chromosome of a eukaryote DNA. We do not find histones in the DNA in the mitochondria and the chloroplast. If we look at the first amino acid made in a eukaryote cell, it's always methionine. In bacteria, it's a similar amino acid, formal methionine, but a different amino acid. And in eukaryote organelle, meaning the mitochondria and the chloroplast, it's always formal methionine. Already mentioned that ribosomes differ. The eukaryote cell grows by mitosis. The prokaryote cell, bacteria, grows mainly by binary fission. And the mitochondria and the chloroplast, they grow by binary fission. So there's some of the evidence supporting the theory that the mitochondria and the chloroplast were at one time bacteria. And then the stronger piece of evidence that the mitochondria and the chloroplast were at one time bacteria is when we sequence the DNA of the mitochondria and the chloroplast. What domain do they fall into? They fall into the domain bacteria. They do not fall into the domain eukaryote, meaning the DNA sequence of the mitochondria and the chloroplast is similar to the DNA of the bacteria. Any question about any of that? All right. So the endosymbiotic theory explains how we got the mitochondria and how plant cells got both the mitochondria and the chloroplast. Plant cells differ from animal cells. Animal cells only took in one bacteria, which became the mitochondria. Plant cells took in two bacteria, probably at different times. The first probably being the mitochondria, and then the second bacteria taken in by the plant cell became the chloroplast. Any question about any of that? All right, that's it for chapter 4. We have a little bit of time. I want to just start with chapter 5. I don't think we need to, but I want to get ahead. So let's go ahead and start chapter 5, microbial metabolism. All right, as is normal, I show you a slide showing you the major goals of chapter 5 and the rough outline. Chapter 5 is on microbial metabolism. Know the terms. What is catabolism, anabolism, activation energy? Know what's an enzyme. What is an active site to the enzyme? What is competitive inhibition? Non-competitive inhibition, also called allosteric inhibition. What is a redox reaction? What is a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph? What is fermentation? And then lastly, what are cyclic and non-cyclic photosynthesis? Any question about any of these terms? Now remember, these are the major goals. To know all the goals, you need to look at the objectives for chapter 5. Also in chapter 5, know what feedback inhibition is. And then lastly, we'll talk a lot about what aerobic respiration is. So understand what it is, know its steps, and how it differs from anaerobic respiration. Any question about chapter 5? All right. When we're talking about chapter 5, we're going to talk about microbial metabolism. You should know that metabolism is all of the chemical reactions in a cell. So all of the chemical reactions in a cell make the cell's metabolism. Because we're largely going to be talking about microbes, where one cell is an organism, we can also say that the metabolism, at least of microbes, is all of the chemical reactions within an organism. Metabolism has two parts, catabolism and anabolism. Catabolism is the making of smaller molecules from a larger molecule. It's a decomposition reaction and it releases energy, meaning we have a complex molecule such as starch. We catabolize it, that gives off energy, to make a simpler molecule like glucose molecules. Glucose molecules have less energy in them than starch, and so this reaction gives off energy. So it's an energy releasing reaction. The main look at catabolism is the size of the molecule. Catabolism is always a complex, larger molecule being broken down into smaller molecules. Whenever you change energy from one form to another, some energy is always converted into heat. Heat is just another form of energy. And this is just the second law of thermodynamics. Whenever you change energy from one form to another, you always have some of that energy converted into heat. In some reactions, some catabolic reactions, we can get energy and take some of that energy to make ATP. This slide is a bit oversimplified because in animal cells, there are only two chemical reactions that can actually make ATP that are catabolic reactions. And that is the aerobic respiration can make ATP. And then the second reaction that animal cells can make ATP from is fermentation. Those are the only two catabolic reactions that animal cells can make ATP. So most of the reactions that are catabolic in an animal cell, such as the splitting of sucrose into glucose and fructose, do not generate ATP. So the vast majority of catabolic reactions in an animal cell do not make ATP. Even in a plant cell, which has several more reactions that can make ATP, or at least a few, most of the catabolic reactions in a plant cell do not make ATP. But they have more than an animal cell. Animal cells only have two. Any question about any of that? All right. We also have anabolic reactions in metabolism. Anabolic reactions or biosynthesis reaction where we take a smaller molecule like glucose and we make a larger molecule such as starch. This reaction needs energy to go. You need energy input into the system. Usually cells get the energy they need to make the biosynthetic reaction by obtaining that reaction energy from ATP. Once again, the second law of thermodynamics states that whenever energy is converted from one form to another, such as the energy in glucose being converted into the energy in starch, and you need energy coming into the system because starch has a lot more energy than glucose. But the point is that some of that energy is converted into heat. It's the second law of thermodynamics. You always get some of that energy being converted into heat. And literally, they say that energy is lost as heat. Heat isn't really losing the energy because heat is another form of energy. Any question about catabolism or anabolism? In the anabolic reaction requiring energy, if energy comes from ATP, ATP is broken down into ADP, an inorganic phosphate, that releases a large amount of energy from the ATP, which is the energy needed by this anabolic reaction to proceed. Let's talk briefly about metabolic pathways. A metabolic pathways is a sequence of enzymatically catalyzed chemical reactions in a cell. Like we can have molecule A, which is catalyzed by enzyme 1 into molecule B. And then you can have molecule B catalyzed by another enzyme, enzyme 2, which is changing molecule B into molecule C. And then molecule C catalyzed by enzyme 3 into molecule D being catalyzed by enzyme 4 into molecule E, and then molecule E being catalyzed by enzyme 5 into molecule F. Molecule A is the starting substrate, what we're starting this chemical reaction with, and molecule F is the end product of the metabolic pathway. So this is the end product, that's the starting substrate. Everything else are intermediate products, and you can call molecule B an end product of the first chemical reaction, but it is the starting substrate of the second chemical reaction. So these are intermediate products, and they're both an end product of the first chemical reaction and a starting substrate of the next chemical reaction. Metabolic pathways are determined by enzymes. Enzymes are encoded by genes, so metabolic pathways come about by the genes in your cells. Any question about any of that? All right, here is a slide showing you all of the metabolism we're going to talk about in this lesson, and you only need to know all of it. Any questions? I'm kidding, you don't need to know all of this. Just showing you that in a single cell, this would be about one cell, there's a huge amount of metabolism going on in a cell. All right, chemical reactions occur by the collision theory. The collision theory states the chemical reactions occur when atoms, ions, or molecules collide with each other. And the chemical reaction will happen if the atoms, ions, or molecules have enough energy in their collision to get the chemical reaction to proceed. The activation energy is the energy needed to disrupt the electronic configuration in the original atoms, ions, or molecules, disrupt them to make the energy configuration seen in the end product of this chemical reaction. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction by increasing the abundance of enzymes. The more enzymes you have, the more likely you'll have a collision between the enzyme and the starting substrate, and then the enzyme will convert that starting substrate into the product, and so we'll speed up the reaction if we have more enzymes. Likewise, you can increase the temperature, and that will increase the reaction rate. If you increase the temperature, the atoms, ions, and molecules will have more movement. They'll have more energy. That's why they have a higher temperature. And that will increase the reaction rate because the more energy the starting atoms, ions, or molecules have, the more energy they'll have in the collision, and that will be more likely to disrupt the electronic configuration needed to perform the chemical reaction. You can also increase the pressure, and what you do is you essentially make the molecules closer together, and when they collide, they have more energy just because there's more pressure. It's a little more difficult to understand, but you increase the pressure, you also increase the reaction rate. Any question about any of that? So enzymes reduce the activation energy of a chemical reaction. Here we have a starting substrate that's going to break down into the products. But before the substrate can break down in the product, the substrate has to, this being the level of energy here, the substrate has to go up to this level of energy, and that's the activation energy required by the substrate to be converted into the product without an enzyme. For example, if I had glucose in this cup here, not glucose, I guess I should say sucrose in this cup here. Yeah, you wouldn't notice difference between glucose and sucrose in the cup. So we'll say glucose in the cup here. You can convert glucose into CO2 and water, and eventually all of that glucose would be converted into CO2 and water because the glucose has more energy in it than CO2 and water. So this reaction is favored, but it doesn't happen very often, would it? Like if I were to look tomorrow, the cup's going to look like it has all glucose in it. It won't look like there's any CO2 and water. In fact, you would need at least a century before you would notice that glucose is starting to get a little liquidy here. You have to have it sealed so you contain the liquid, the water in it. But my point is, eventually, all of the glucose would turn into CO2 and water, but it's not going to happen anytime soon. Probably not in our lifetime. And that's because the glucose needs a high activation energy without an enzyme to be converted into the CO2 and water. I could get it to happen immediately just by touching a blowtorch to the glucose, and then that's rising the temperature to either 1,000 degrees Fahrenheit or 1,000 degrees Celsius. I don't remember which. Doesn't matter. It's very hot, and that will ignite the glucose, converting it into CO2 and water. I'd have to step away because so much energy would come out of the glucose, I couldn't be anywhere near it. That differs from glucose being converted into CO2 and water by an enzyme. And that happens in your cells all the time. What temperature is your cells at? It's not 1,000 degrees Fahrenheit or Celsius, is it? So what temperature are your cells at?
98.6?
Yeah, 98.6 or 37 degrees Celsius. That's quite a bit different than 1,000 degrees, either Fahrenheit or Celsius. I don't remember which. Doesn't matter. It's quite a bit different. And glucose in your cells is converted into CO2 and water all the time. What happens is the enzyme can break down the glucose at a much lower temperature, 37 degrees Celsius, breaking it down to CO2 and water, showing you the difference in the activation energy with an enzyme and the activation energy without an enzyme. Any question about any of that? All right, I think I'm going to stop here, and we're going to stop the lesson early, and at 6:30, I will continue with the lab. Any questions? All right. I'll see you at 6:30.